Hello, my name is Hannah Gorman, and I will be discussing interferon alpha 2. This is the primary structure. You can see from the amino acid sequence that it has 188 amino acids, and here we can see some of that composition, what some of those specific amino acids are. There's hydrophobic ones. There's cysteine, which we know will form disulfide bridges, but we can't tell where those will be yet, and there is also lysine, which can form salt bridges. So the secondary structure, we can tell from this 3D representation that there's a lot of alpha helices. Those are held together by hydrogen bonds between the carboxyl and amide of the individual amino acids. And we can also tell this represents how there are hydrophobic amino acids about every third or fourth. So we can tell this is this alpha helix right here from amino acids about 111 to 132, that there will be a hydrophobic face on this alpha helix, and that will have an important role in the tertiary structure. So here's the tertiary structure, like I was just talking about. So there's hydrophobic and hydrophilic faces of the individual secondary structural components, and those will participate in the hydrophobic effect, so that the hydrophilic regions will face externally towards the aqueous environment, and the hydrophobic ones will face internally, making the protein more energetically stable. We also know from those cysteines that we saw in the primary structure that there will be disulfide bonds. So here's an example of one of those disulfide bonds between an alpha helix and a beta strand. And we also know that there will be salt bridges between charged amino acids. Here is the tertiary structure. Again, this is just the kind of bringing that all together. So this is the signaling region of the protein. It's this amino acid right here from amino acids 1 to 23. And here is this alpha helix specifically. Within it, we can see a lysine, which is charged and we know can form salt bridges. So we know that this could potentially be a signal to other molecules that this particular region exists on this interferon alpha 2, and that could play an important role, especially this being a ligand in its function as a signaling molecule. So we also know that it has a receptor region, or it has a binding region, and that's where it binds to the interferon receptor, and that region is this alpha helix here, here, and here, these three, which are from amino acid 155 to 100. And we know that that binds selectively and non-covalently, so that could be in the form of a salt bridge. And so we do know that there's a glutamate right here, and that could form a interaction with another charged particle to form a salt bridge and indicate to the receptor that this ligand is present and set off a signaling cascade to warrant a cell response.